Hello everyone, I welcome you all to Biology Lessons with Kalyani. In today's chapter, we will be discussing about the part 2 of the chapter Plant Growth and Development. In part 1, we already looked into what are the different plant growth regulators or phytohormones, and in this chapter, we will be looking at what is plant growth and what is plant development. So let's see what is plant growth first. Plant growth, it is one of the most fundamental and conspicuous characteristic, right? A plant is growing. You can make it out easily, right? It will grow, it will become tall, or it will have more number of leaves. So it is a very obvious process. Now, what does this mean? Plant growth refers to the increase in the protoplasm of a plant cell. Consider this as a plant cell. Now the protoplasmic content increases in this plant cell. That refers to be as a plant growth. As a result of increase in this protoplasm, the size of the cell increases, volume increases, weight increases, and after reaching a certain size, the number of plant cells also increases because the cell tends to divide, right? So plant growth, it is the irreversible and permanent increase in the size, volume, or weight of the plant parts such as plant cell or the different organs of a plant, right? Plant growth also refers to the increase in the number of plant cells, right? So that is plant growth. In this process of plant growth, we have both anabolism and catabolism because the food is broken down and it is absorbed to make the protoplasmic content. But the rate of anabolism is more than that of catabolism in case of the plant cells, right? So plant growth overall can be considered as a metabolic process having anabolic rate more than the catabolism. The important characteristics of plant growth are it is indeterminate in nature. What is determinate? Determinate means something which is limiting. But plant, if you see, plants can grow throughout their lifestyle. Throughout their life cycle, they are capable of growth. They can produce new leaves. They can produce new branches. So that type of growth is called as the indeterminate growth. Also, in case of plants, new cells are always added in their body, right? So that form of growth is called as the open cells. Now, how this indeterminate and open type of growth form is shown in the plants? It is because they have some specialized cells in them which are called as the meristematic cells that are always capable of dividing and forming new cells, right? So this is your plant growth. Now let's see what is plant development. Plant development is nothing but as a fertilized egg, zygote, or a seed, plant seed, it germinates in the favorable condition, and a series of highly ordered structured events that take place leading to the formation of the mature plant which has complex structural organization such as leaves, branches, roots, stem, fruits, flowers, seeds, right? Which each of these parts they perform specific function or are differentiated. Differentiated means nothing but unspecialized cells performing specialized function. So these series of events which change a plant zygote or fertilized egg or a seed to a mature plant until again to its death, all these events together they are known as development, right? So essentially in plant development we will be having growth of the plant cells, that is increasing protoplasmic content, as well as the cells they become differentiated or assume specialized function. So development can be referred to be as plant cell growth plus differentiation. right? I hope this part is clear. Now let's look into the plant growth nature, which is indeterminate and open. I already told that plant cells are capable of growth throughout their life cycle, right? That which is called as the indeterminate growth. And open type, why? Because the new cells are added in a plant always. Now this is possible because plants have specialized cells called as the meristematic cells, which have the capacity to divide and self-perpetuate. That is, they can always keep on dividing and forming new cells. And the product of these meristematic cells often lose the capacity to divide, become specialized and perform specific function, right? So these meristematic cells, which are responsible for the indeterminate and open type of growth in plants, they can be of two types: primary or early meristem, lateral or secondary meristem. These primary meristem, they are present in the early life cycle of a plant and comprise of ram, which is a root apical meristem, and stem, which is a shoot apical meristem, right? Now see, the uh, this is the structure of a plant, and in the shoot apex you have shoot apical meristem, which leads to the formation of the cells. See, the cells are formed downwards, new cells are being added downwards, and root apical meristem, it is formed on the base of the plant, and it is responsible for adding new cells in the upward direction. Here the direction in which new cells are added is very important, and you have to pay attention to it. Okay, now these ram and sam, essentially what are they doing? They are adding new cells along the vertical axis, so they are responsible for the elongation along the axis, right? Now let's come to the lateral or the secondary meristem, which is developed in the later stages in case of the plants. These lateral meristem, this comprises of the vascular cambium and the core cambium. Now these two meristematic cells, uh, they are responsible for increasing the number of the cells in a horizontal direction or lateral direction, right? So they are responsible for increasing the girth of the plant. Is that clear? Let's now see how we can measure the plant growth. We just discussed that the plant growth, it refers to the increase in the protoplasm, but the increase in protoplasm, it is uh, in the new cell. To take out the protoplasm and to measure it, it is a very difficult task. So we devise some indirect mechanism. The parameters which are also proportional to the increase in protoplasm, those we consider for measuring the growth of the plant. For example, we can take fresh weight, which is nothing but directly we take a plant sample and we measure its weight. And dry weight, after drying the plant sample, if we measure the weight, that is called as the dry weight. So this uh, increase in the fresh weight or the dry weight, it indicates that growth has taken place in the plants, right? For some plants, we can also take for some plant parts, we can also take the measurement of the plant growth in terms of the increase in length, especially if we take pollen tube. Now pollen tube always increases in the length in the vertical direction, so if there is an increase in length in the pollen tube, it indicates that plant growth has taken place. Similarly, if we consider the leaves, In case of the leaves, if you consider a young leaf, it will be very small in size. As the leaf grows, the surface area of the leaf increases. So the increase in surface area of the dorsiventral leaf can also be taken as a measure of the plant growth. Similarly, increase in volume of the cells is also taken as a measure to find out the plant growth. Now, sometimes we can also take increase in the number of cells. For example, if you consider maize, one cell of the maize root apical meristem, it is capable of adding 17,500 new cells per hour. So, as the number of cells are increasing, the growth is taking place in the plant. Right. Similarly, if we consider watermelon, here we can consider the plant growth with respect to size. Watermelon, it increases in size about 3,50,000 times. So here increase in size is happening, and it is indicating increase in protoplasm or the plant growth. So all these different parameters, fresh weight increase, dry weight increase in fresh weight, dry weight, length, area, volume, cell number and size, they can be taken as parameters to find out the plant cell growth. Is that clear? After we have seen how we can measure the growth, let's see different phases of the plant growth. Now this is the diagram of a seedling. This is its root portion. Now if we put marks after the seed grows, there is elongation in the root, and if we draw parallel lines, we get different zones. We get zone A, in which there is a little increase in length, and zone B, C and D, in which the increase in length is very evident, and there is no or very minimal change in the length of the bands of C, of E, F and G. Okay, so we can divide the plant growth into three different phases as per this, as per the increase in the cell length or the increase in the elongation of the cell. The first band, that is in the bottommost part, that is the meristematic phase. So in this stage, what happens, the meristematic cells are produced. So meristematic cells, we all know, they are capable of dividing continuously. They continuously form new cells. So these are present both in root apical meristem as well as shoot apical meristem. Because these plant cells, these meristematic cells, they divide very rapidly. They need to be rich in protoplasm. Also, they should have large conspicuous nuclei, because we know once a cell is dividing, the nucleus should be very prominent in them, and thin cell wall should also be present. Why? Because thin cell wall, if the wall is thick, it will hinder or have obstacle in division process. If the wall is thin, so easily the cells can divide. Also, there is abundant plasmodesmatal connection. Why abundant plasmodesmatal connection required? See, plasmodesmatal connection, they are the cytoplasmic connections between the cells. As the cells are growing, they grow together. So this plasmodesmatal connection, they help in signaling exchange of the molecules, so the growth can take place in a very ordered way. So the characteristics of the meristematic phase are cells rich in protoplasm, conspicuous nuclei, thin cell wall and abundant plasmodesmatal connection. Right? The cell undergoes very less elongation in the meristematic phase. Now let's come to the second phase, that is the zone of elongation. Where is the zone of elongation located? The zone of elongation, it is located proximal to the meristematic zone. This is your meristematic zone or the meristematic phase. in the proximity or near to this, we have the zone of elongation, and it is away from the apex. And the characteristics of the zone of elongation are the cells will be very elongated because the cells are trying to get bigger in length. That's why we will have elongated cells. There will be new cell wall formation and deposition of new cell wall material, and also we will get abundant vacuolation or increased vacuolation. Now here it is important why we will have more vacuoles. It is because as the cells need to grow, it needs to have more turgor pressure, and this is possible only when the cell has more water. And water, where is it stored? It is stored in the vacuoles. So in the cells in the zone of elongation, they have increased vacuolation. The last part is the zone of maturation. In this zone, the cells have already attained the maximum size or maximum elongation. They have also undergone maximum thickening, and all the protoplasmic modification necessary has already taken place in the cells of zone of maturation. So they are all matured and they are having specific function. So there is, if you see, the length of the cell or the dimension does not change much in the zone of maturation. I hope you are clear about the different phases of the plant growth. So until now we saw what is growth, how we can measure plant growth, what are the different phases of plant growth. Let's now see what is plant growth rate, how we can measure the growth rate. Okay, how we can define growth rate? Plant growth rate can be defined as increased growth per unit time. That is referred to be as growth rate. Mathematically, we can represent it as increase in growth per unit time. And we can have two types of plant growth rate, arithmetic and geometric. In arithmetic, what happens, suppose this is the first cycle of division. Okay, this is your cell which is undergoing division. Now it will form two cells, and among the two cells, one of them will have the capacity to divide, the other will not have the capacity to divide. So this, in second, we will have how many cells? Two. We started from one cells, then we have two cells, and then again if you see third division, so we will have one cell which will have the capacity to divide, all others will not be able to divide. Okay, then again if you see fourth cycle of division, so this one will be forming one cell which can divide, and then we will have total of, here we have three cells, and then we have four cells. So if you see the number of cells, how is it increasing? Is it increasing arithmetically or not? First we have one cell, two cell, three, four, five, etc. So this type of growth is called as the arithmetic. This type of cell growth is called as arithmetic growth rate. Is that clear? Now let's see the second type of growth rate, that is a geometric growth rate. Here what happens, all the cells, they take part in the cell division. So let's say we started with one cell. Okay, so this is the starting cell. Now it will form two cells. Okay, now these two cells can again form another two cells, and this one will also form two cells, right? This one will again form two cells, then two cells. So if you see, if in the beginning we had one cell, then we have two, then we have four, and then we have 1 2 3 4 5 6 7 8, so we have 8 cells. So the increase in number of cells is taking place in a geometric fashion. Okay, So this is in a geometric series. That's why this growth rate is called as the geometric growth rate. Okay, in this figure also, this deep blue, this represents the cells capable of division, cells that lose the capacity to divide. See, only one cell is retaining the capacity to divide and form new cells, others just they are remaining intact, right? Whereas in geometric, all the cells take part in division. So if you see a zygote, when a zygote divides, the division takes place in the geometric phase, because all the cells take part in the cell division process, right? So when we see arithmetic growth rate, in case of arithmetic growth, the when the cells divide, only one among the two daughter cells, it continues to divide, while the other, it differentiates and matures. Other does not undergo cell division. Example of this can be root elongation at a constant rate. Now, if we have to mathematically represent it, it can be represented as LT is equal to N naught plus RT. Now, if we consider the root elongation, so LT is the length at time t and L naught is the length at time 0. Now, if you see at time 0, it will be having some length. So, this is nothing but L naught. Now, after time t, the length is increasing in an arithmetic way, in an arithmetic progression. So it is increasing at a constant rate, so it is given by LT is equal to L naught plus RT. Now what is R? R is the growth rate or elongation per unit time, and T is the time until which the elongation is happening. So when we plot the graph, the height of the plant and the time, we will get a straight line, right? Is it clear? I hope the arithmetic growth is clear. Now let's see what happens in the geometric growth. Now we saw that in case of geometric growth, the progeny cells, the which are obtained from mitotic cell division, all of them, they will be dividing and forming daughter cells. So what will happen when we start from the initial time? So this is time, this is in the y-axis we have the size or the weight of the organ, right? Initially the growth is slow, and slowly we will have a lot of cells, and each of the cells will be dividing, so we will be getting a rapid growth phase, or the exponential phase, or the log phase. Then what will happen after some time, or the saturation time, the nutrients will be limiting. So in this situation, the growth will tend to slow down or become stagnant. That is called as the stationary phase. Is that clear? Initially, it will have a slow or the lag phase, then rapid or exponential phase, followed by a stationary phase, in which we will be having the limited nutrient supply. Mathematically, we can represent, if we see weight T or W1, that is weight at time T is equal to W naught, or the initial weight, into ERT. E is nothing the base of natural logarithm, and T is the time for the growth, R is the growth rate, and W naught is the initial size in the beginning of the period, right? This R is also called as the relative growth rate or the efficiency index, and W1, it depends upon the initial weight of the plant sample. Is that clear? Or initial weight of the organ. So this geometric growth, it shows a typical S curve or a sigmoid curve. I hope this geometric growth is clear for you guys. Let us now see how we can quantitatively compare the growth. Okay, suppose we have two leaf samples. One is 5 centimeters square, surface area, other is having 50 centimeters square surface area. So we will be taking dorsiventral surface area. Now, after time t, we found out that the surface area increased to 10 centimeters square, and in the plant B, it increased to 55 centimeters square. Now, if we see the absolute value, the 55 centimeters square area is more than 10 centimeters square. So if we just consider the absolute value, we can say the growth is more in B compared to A. But we should we also have to consider the initial leaf surface area. So we also have to take that into account. So if we take that into account, so what will we have? We can see that for A, the initial area was 5 centimeters square. After time t, we got an area of 10 centimeters square. So if we find the relative rate, we will be getting 2 times. So the relative growth rate is 2 times or twofold. But if in case of B, if we see, initially it was 50 centimeters square area. After time t, it became 55 centimeters square. So the increase in the area, leaf surface area, is 1.1 times or 1.1 fold. So the relative growth rate, if we compare, so the relative growth rate of A is more than that of the B. So the growth rate in case of A is more than that of the B, right? I hope that is clear for you guys. If we see plant growth, it depends upon many different conditions. The most important among them is water. Why water is very important? Because it is the main medium in which all the enzymatic activities take place. And secondly, increase in size, enlargement of the cell, extension of the cell, all of them means that the cell need to be turgid, or the protoplasm content is increasing, and it directly means that the water content or the water status is increasing, right? So water is very essential for the growth and development of the plants. That's why we are advised to water the plants on a regular basis, or the plants will have wilting or sometimes death in extreme conditions. The second important element which is important are the nutrients. The elements which are required in less quantity, they are called as the micronutrients, and those required in more quantity, they are called as the macronutrients. Nutrients are essential because they can act as source of energy. Also, they are required for the synthesis of the protoplasm. Third important condition for growth is oxygen. Why oxygen is essential? Because oxygen helps in carrying out the respiration and hence releasing the metabolic energy. And we know respiration needs a lot of energy. That's why we know that plant growth needs a lot of energy. That's why oxygen is very essential for plant growth. And plants also have an optimal temperature range in which optimal growth can be taken place. Other important factors are light or photoperiod and gravity. These environmental signals play important role in the different stages of the plant growth, right? So in last slide, we discussed what are the different elements required for the plant growth. Now these plants, they are very fascinating organisms. They show some very special characteristics, which is known as differentiation, dedifferentiation, and redifferentiation. Let's look into what each of them means. Okay. Now, plants we have already discussed, they show an indeterminate and open type of growth. Why? Because they have special cells called as the meristematic cells. Example, RAM, SAM and cambium cells. Now, these meristematic cells, if we take, they are unspecialized. They do not have specific function. But as the plant grows, the meristematic cells, they mature, they undergo structural change in the cell wall as well as the protoplasm, and they differentiate and perform the specific function. This entire process in which the unspecialized meristematic cell, they become specialized, it is called as differentiation. That is, the cells attain specificity in the stage of differentiation. One example we can take for this is development of the trachea element from the meristematic cell. Okay, now see, consider this is the meristematic cell. It is having a conspicuous nucleus. We already discussed meristematic cells have a big prominent nucleus, small vacuole. Okay, it is having a small vacuoles. Now what happens? We know trachea elements. Tracheates are elongated cells and required for the water transport, right? So here what happens, this meristematic cell undergoes a series of change. What are the changes? The prominent nucleus becomes less prominent, and finally it is not visible only. The cell, it becomes elongated, and the protoplasm, it is lost. There is loss of protoplasm. Why? Because it is designed to carry the water, and the water, as it moves from the soil to the plants, it will have a very high water tension. So in order to sustain that tension, the walls of the trachea element should be very thick, or it should have a, that is why what happens, the meristematic cells, they lose their protoplasm, change their structure, that is, they become elongated, develop secondary walls, thickening like lignocellulosic walls they will have, and they have thicker walls so as they can sustain the water tension. Okay, and then finally they assume the shape of the trachea element that that looks like elongated, that is, looks very elongated. So this process of unspecialized meristematic cell becoming trachea element, it is called as the process of differentiation. Is that clear? Now plant is so interesting. See what happens sometimes in the life cycle of plant. What happens, there will be situation in which these differentiated cells, they need to become unspecialized. So the process in which this differentiated cell act like meristematic cells and become unspecialized, that is called as deep differentiation. Okay, this de-differentiation or loss of differentiation, it takes place in the secondary stage of the plant life cycle. So where does it exactly happen? It takes place in the formation of interfascicular cambium and the core cambium. How it takes place, let us have a look. So see, this is a cross section of the plant. This is the xylem. Blue ones is the xylem, red one to the outside is the phloem, and in between the xylem and phloem we have the intrafascicular cambium. So you can see star marks, purple stars I have marked. So what happens, these are the differentiated parenchymatous cells. Okay, sometimes what happens, these parenchymatous cell during the secondary growth stage, they lose their specificity and tend to become unspecialized or begin acting like the meristematic cell. So see, these parenchymatous cell tend to form a continuous ring with the intrafascicular cambium, and they form the cambial ring. So this is the this is called as the secondary meristem or the interfascicular cambium, because it is between the two fascicles. That's why it is called as the interfascicular cambium. Is that clear? So this process is called as de-differentiation. There is another process which is called as redifferentiation. what happens here? It is very interesting. See, the so we had parenchymatous cell which were specialized. They became despecialized, or they became unspecialized and formed the interfascicular cambium. Now this interfascicular cambium is type of the unspecialized cell, right? This purple. Now what happens? It can form specific cells having specialized function, that is secondary phloem and secondary xylem. Now this is called, this formation of specialized structure from the dedifferentiated cell, which have already lost the capacity to divide, which don't have the capacity to divide any further and become mature and perform specific function, that is called as the redifferentiation. Is that clear? It means the secondary meristematic cells or the unspecialized cells again form the specialized cells which have specific function. Okay, example is formation of secondary xylem and formation of secondary phloem. This entire process is called as redifferentiation. This entire topic is very important from exam point of view. So we already saw that growth is in plants is open. It can be indeterminate or determinate. Now even if we see that differentiation, if we see differentiation in plants is also open, that means we have a lot of choices. How? Because the final structure, what a meristematic cell is going to form, that depends upon the location of the meristematic cell. For example, the cells, if they are positioned away from the root apical meristem, they differentiate in the root cap cells, and the cells which are pushed to the periphery of a root, they form the epidermal layer. Is that clear? So we can say that differentiation in case of plants is open. There is another interesting phenomena which takes place in the plants, that is called as plasticity. We all know plastic, it means that something which can change, right? In case of plants, ability of them or ability of the plants to show different structures or different phenotypes or change in structure or phenotype of a plant with response to the environmental condition or in different phases of its development is known as plasticity, right? That is, if a plant is showing different phenotypic structures in its life cycle or in response to the environment, we call that term plasticity, right? If the plant is showing different leaf structures in different phases of life, that is known as developmental heterophylly. Examples of them can be cotton, coriander and larkspur. Here the leaf structure of larkspur is given. This is a juvenile or the young leaf of larkspur. See how it is looking, and when it is adult, the leaf is looking different. So it is showing two types of leaf structures in the different phases of the life. Young leaf is different or juvenile leaf is different from that of the adult leaf. So this term is called as developmental heterophylly. And second is your uh, in response to environment, that is called as environmental heterophylly. So see, example of it is buttercup. Now buttercup, you can remember it with a cup which is used to drink water. So this buttercup, it will have two types of leaf, aerial or terrestrial leaf, and aquatic or aquatic leaves. Now, the aquatic leaves of the buttercup which lie inside the water, they are finely dissected. Why? Because thin leaves increases the surface area and hence increases the gaseous exchange. Now, if you consider the terrestrial leaves or the aerial leaves, they are broad, they are lobed leaves, and this helps them to capture more sunlight, right? So these two types of leaves that are present in buttercup in both in the land and water is an example of environmental heterophylly. It's a very important concept. Often questions come that cotton, coriander, larkspur, example of which heterophylly? Developmental heterophylly. And buttercup, example of environmental heterophylly, right? I hope this is clear now. Let's move on to the next part, that is development. We already have discussed that development refers to all the changes that a plant or any organism undergoes throughout its life cycle. That is, from germination until the death of the plant, whatever it changes it undergoes, it refers to development. So essentially that means in development will comprise of growth or increase in the protoplasmic content, as well as differentiation, that is specialization of the plant cells to perform specific function, right? Now this development, it depends upon two types of factor. It can depend on the internal or the intrinsic factor, or external or the extrinsic factor. These intrinsic factors on which plant development can depend are intracellular or the genetic factor, or it can also depend upon the intercellular factor, that is a plant growth receptor or plant growth hormone, right, or the phytohormone. We already have discussed about it, and we know the how the genes that can determine the plant growth. Okay, and the plant growth hormone, we have discussed all five important classes of hormones, right, how they can if have an impact on the plant growth. The extrinsic factors of the plant also we have discussed, the important elements required for growth, same, light, temperature, oxygen, nutrients, gravity, all these will be considered under the extrinsic factors. Okay. So this is the schematic representation of development process in plant cell. See how the meristematic cell, which is unspecialized, how it is undergoing increase in the protoplasm content, or undergoing plasmatic growth, or cell growth, and then it is undergoing differentiation, which comprises of elongation as well as maturation, and these mature cells perform different structures. Eventually the plant undergoes senescence and then death. This entire process is together called as a process of development, right? So you must be clear with the two words now. Growth is the increase in protoplasm content, whereas development is the entire stages throughout the plant's life cycle. I hope all the concepts here in plant growth and I hope you are clear with the plant growth and development chapter. Today we covered what is plant growth, what is plant development. In the part 1 of this video, I have discussed about all the important plant growth hormones or plant growth regulators. If you have not referred to this video, please find the link. I'll be attaching it here and refer to that. Also, I will be uploading a keynote video which will comprise of all the important points in short, which you can refer as a for your revision purpose. Okay, if you like the video, please like, share and subscribe to my channel. And yeah, I could not upload videos shortly for a couple of weeks before because I was not keeping well. I had a very bad throat infection, but I'll make sure I will be putting videos regularly. Okay, so thank you everyone. Have a very good day.